Welcome in the class. A very good afternoon to everyone. Let's crack CSIR UGC NET and GATE 2021 together. And that is the name of our channel. That is Let's Crack CSIR UGC NET. Shona is in class and she's saying, "Hi ma'am." Hello Shona. You're watching this video under the platform of India's largest learning platform, that is Unacademy. I'm your educator Farida Johar Bandukwala. I have teaching experience of 10 years. I've qualified MP SLET examination in year 2018. You're going for an academy board, all things you are going to get. You are going to get daily live classes. That is every day you will get to attend the classes so that the momentum of study would be there. Then live class is going to make you ask your doubts and queries while the class is going on. The whole syllabus is quite structured so that you can complete the whole syllabus in a small span of time and in a very structured manner. And then live tests and quizzes are taken so that one can evaluate that how much he has learned so far, and unlimited access towards all the videos whichever are recorded in that particular category. An academy tries to do the live classes at two places, that is YouTube. Presently you are watching a YouTube free class, and for watching the YouTube free class you just need to go to the channel, subscribe the channel, and you can hit the bell icon for getting the notification, and you can go and attend the classes, and you can also attend the previously recorded classes. Then there are special classes. For special classes you need to download the Unacademy learning app. After downloading the Unacademy learning app, you need to make your account. Then you will be asked to fill the referral code of an educator. So fill my referral code, that is FB01. By filling my referral code, you will be getting an additional 10% discount in the Plus subscription. And for free class, you can watch it completely free of cost. Then there is this Plus subscription, and this Plus subscription is a paid subscription. You need to pay a certain amount, and for that you need to pay a amount that is quite pocket friendly. But Plus subscription contains all the units in a very systematic way, and all the points are covered, topics are covered. And other than that, it's a on this platform short-term courses, crash courses are continuously conducted for all the upcoming classes, so that you can always have that mode of learning, that tempo of learning. And after that, use my referral code for getting an additional 10% discount in the Plus subscription. So my referral code is FB01, so do not forget to use my referral code FB01. So this was a little introduction. Now moving towards the topic of today, that is YAC, or you can call it yeast artificial chromosome. Yeast artificial chromosomes. Yeast artificial chromosomes. Now in yesterday class we did vectors, and in vectors we have seen one type, that is plasmid. Now when we are talking about yeast artificial chromosome, it is also a kind of vector. So we are just continuing the yesterday's topic, that is vectors, and now we are seeing another type of vector, that is yeast artificial chromosome. So the topic under which yeast artificial chromosome comes under is vector. It's a kind of vector, right? So yeast artificial chromosome is basically a human engineered DNA molecule, and it is used to clone the DNA sequences in the yeast cells. So we'll write the sentence that yeast artificial chromosome is a human engineered, the human engineered DNA molecule, and it's used to clone DNA sequence. And where? DNA sequence in yeast cell. In yeast cell. So this is important. So what you need to remember in this page is that it is used for cloning in the yeast cell, otherwise human engineered. Okay, most of the vectors which we use are almost human engineered, so it is used for cloning in yeast cell. So we'll just rub this and we'll write a simple one line over here, used for cloning in yeast cells, in yeast cell. Right now, this yeast artificial chromosome is a plasmid shuttle vector. The YAC is a plasmid shuttle vector. It's a plasmid shuttle vector, and it is capable of self-replicating and being selected in common bacterial hosts such as E. coli, as well as in the budding yeast cells, that is Saccharomyces cerevisiae. So when we are saying that it's a shuttle vector, YAC is a shuttle vector. So it is a shuttle vector between what? E. coli and Saccharomyces cerevisiae. So capable of replicating in E. coli and Saccharomyces cerevisiae. So this is the thing, that it can replicate in both of them, that is in E. coli and Saccharomyces cerevisiae. Now talking about yeast artificial chromosome. Yeast artificial chromosome is a artificially constructed chromosome. So what all things it contains? So as the name is saying that it's a yeast artificial chromosome, so it's a artificially constructed chromosome. It's a artificially constructed chromosome containing what all things it is going to containing. If it's a chromosome, it should contain a centromere. So yes, it has got a centromere. A chromosome has got a telomere. So yes, it has got telomere. Then it has got autonomous replicating sequence element required for the replication and preservation of yeast artificial chromosome in the yeast cell. So autonomous replicating sequence, we'll write the full form. Autonomous replicating sequence element, and it is required for replication and preservation of yeast artificial chromosome. So why is it needed? That also we'll write. Needed for replication and preservation of yeast artificial chromosome. After that, ARS elements are thought to act as replication origins. ARS elements are thought to act as replication origin. Replication origins. Now these yeast artificial chromosome or anything whichever we usually talk about history. So yeast artificial chromosome was actually first time described in year 1983 by Murray and Sotak, or Sotak we can say. Some of the words are silent because it belongs to some non-native people. So first described in year 1983 by Murray and Tack. So remember that who gave this yeast artificial chromosome? So in very many local examinations, PSCs, it is asked. Now after that, yeast artificial chromosome, if it contains centromere, it contains telomere, it contains ARS, so it behaves like a normally existing chromosome. But for existing as a normal chromosome, they should have proper size and showing the comparable stability. So we'll write that yeast artificial chromosome behave like naturally existing chromosomes, provided that they are provided that they are of proper size and stability. Right, so they behave like a natural chromosome, that is okay. One thing, naturally existing chromosome. Another thing with it is proper size and stability. Proper size and stability. Now if we are talking about yeast artificial chromosome or anything, we should know that what is the purpose behind of making such thing. So purpose of yeast artificial chromosome is cloning vehicle. It acts as a cloning vehicle. Acts as cloning vehicle that propagate in eukaryotic cell host as eukaryotic chromosome. So propagate in eukaryotic cell hosts as eukaryotic chromosome. So both of the thing can be eukaryotic, like yeast, budding yeast, that is Saccharomyces cerevisiae. So it can propagate in eukaryotic cell host as eukaryotic chromosome, as easy as this. Now clone of very large inserts of DNA can be made. Clone very large DNA inserts, that is 100 kb to 10 MB. Very important that very large DNA inserts can be cloned with the help of yeast artificial chromosome, that is 100 kb to 10 MB. So this was the purpose. Now talking about the features. So features of yeast artificial chromosome. Features of yeast artificial chromosome. So yeast artificial chromosome are kind of cloning vehicles, and they are like plasmids. Cloning vehicles like plasmids. Then finally, chimeric DNA is a linear DNA molecule with telomeric ends in the artificial chromosome. Finally, chimeric DNA can be obtained where we have inserted the gene of interest as well. So final chimeric DNA, chimeric or recombinant DNA, final chimeric DNA is a linear DNA molecule, with telomeric ends, but it is an artificial chromosome. Artificial chromosome, right? So it's an artificial chromosome, and It exists like that. Now, if it is amplified in E. coli and if it is amplified in yeast, because we have seen that it's a shuttle vector. So, what all things you should know when it is amplified in E. coli and what all things you should know when it is amplified in yeast. So, we'll be making two categories. So, amplified in E. coli, amplified in E. coli, and here amplified in yeast. Two things. So when it is amplified in E. coli, it is of relatively small size, approximately 12 kb. Relatively small size, approximately 12 kb, and it is circular form. Circular form. When it is amplified in yeast, it is very large. Several hundred kilobase pairs, approximately hundreds of kilobase pairs. And when it is amplified in yeast, it is also in linear form. Is in linear form. So when it is amplified in E. coli, it is relatively small in size rather than when it is amplified in yeast. These two things you should remember. Now, yeast artificial chromosome plasmids, we'll be talking about that. Yeast artificial chromosome plasmids. So, yeast artificial chromosome plasmids. It will be written as this, PYACs. Okay, so we'll be talking about this. Let us take another color. Yeah. So there are many different yeast artificial chromosome plasmids which exist, and they are in the name like PYAC3, PYAC4 plasmids. So there are many different plasmid or yeast artificial chromosome plasmids which exist, such as PYAC3 and PYAC4. These are the examples. Then the basic structural feature of yeast artificial chromosome were developed from the yeast chromosome shuttle plasmid series. Okay, so they are composed of double-stranded DNA. Then they include the the autonomous replicating sequence of yeast, right? So the basic structural feature of YAC were developed from yeast artificial chromosome shuttle plasmid. So basic structural features of yeast artificial chromosomes were developed from yeast centromere shuttle plasmids YCP series, and these are composed of what are they composed of? They are composed of double-stranded circular DNA. Double-stranded circular DNA sequence carrying the beta-lactamase gene, carrying beta-lactamase gene, and it is abbreviated as BLA, and the bacterial origin of replication, and bacterial origin of replication, and it is written like this bacterial origin of replication. So this is the beta lactamase gene, and this is the for origin. Now include the yeast autonomous replication series 1 with its association with centromere 4 DNA sequence as well as URA3 selectable marker. So what all it contains? Include yeast autonomously replication sequence 1 with its associated centromere 4 DNA sequence as well as URA3 selectable marker. URA selectable marker. After that, the yeast H1S3 is flanked by a telomere-like DNA sequence that are adjacent to the two recognition sites for BAM-H1 and restriction enzyme. So, yeast H1S3 is flanked by a telomere-like DNA sequence. By a telomere-like DNA sequence and contains the recognition site for restriction enzyme. And which restriction enzyme we are specifically saying? BAM-H1, and contains recognition site for BAM-H1. Then most of these yeast artificial chromosomes also contain the cloning site in the middle of the suppressor of an occur allele of a tyrosine transfer gene. So that is also one of the important factor when we are talking about yeast artificial chromosome. So let us make a circular map of plasmid vector of yeast artificial chromosome. Circular map of plasmid vector PYAC3. So it will be like this. So this is the ARS1 and centromere 4. So this is ARS1 and centromere 4. Then so this is SUP4 occur. This is SUP4 occur. Then there is the sequence URA3, URA3. Then there is this telomere here and here. So here telomere. Then there is the site for BAM-H1 that I was telling you. So it contains this BAM-H1 site near the H1S3. So here is the H1S3. So this is H1S3 and here is a site for BAM-H1 and here also it contains the site for XHO1 and 3, right? So these are the sites, and same thing will be there for the sites in the opposite direction. Here there is a blah gene, blah, and again there is this gene, and it would be labeled like this. This is PMB1 uricide, PMB1 uricide, and here it contains one restriction enzyme site. Here it contains, here it is BAMH1, and here it contains for hint 3 and XHO1. Like this is the circular map of a plasmid vector. So the gene is going to run this one in this direction. This runs like this runs like this runs like this runs like this one runs like this, and this one runs like this, So this is the whole circular map of plasmid vector of yeast artificial chromosome. Now we will be seeing construction of yeast artificial chromosome. Construction of yeast artificial chromosome. Just a second. Just give me a second. Sorry for the disturbance. Now we'll be talking about construction of yeast artificial chromosome. Construction of yeast artificial chromosome. So first step is plasmid DNA purification. Plasmid DNA purification. After this, it should be treated with enzymes. So treatment with restriction enzymes. So treatment with restriction enzymes. After this, ligation and yeast transformation. Ligation and yeast transformation. So this was the construction of yeast artificial chromosome. So yeast artificial chromosome is built using an initial circular plasmid, and it is typically broken down into two linear molecules using restriction enzymes. Then DNA ligase is used to ligate a sequence of gene of interest between the two linear molecules, forming a single large piece of DNA. So we are just going to write this also. Let us write it over this side. Just a second. So construction of yeast artificial chromosome, be writing over here that a yeast artificial chromosome is built on or built using an initial circular plasmid. Then typically it is broken down into two linear molecules. Broken down into linear molecules using restriction enzyme. Then gene of interest is inserted between these two linear sequences by DNA ligase. And like this, a large linear piece of DNA is formed. Piece of DNA is formed. Now, talking about the insert, let us deal that how this insert is going to behave. So this inserted gene actually compensates for mutations in the gene host cells, and that causes the accumulation of red pigment. So the host cells are normally red, and those transformed with yeast artificial chromosomes would be actually colorless, the place where it has been inserted. So cloning of a foreign DNA fragment into yeast artificial chromosome causes insertional inactivation of the red color gene, and therefore the colonies that containing the foreign DNA fragment are red. The yeast artificial chromosome vector is capable of carrying a large DNA fragment, and transformation efficiency is very low, to be very specific when we are talking about yeast artificial chromosome. So those all points which I have told you, let us just make a list of this, that this inserted gene compensates for a mutation in yeast host cell that causes red pigment. So the host cells are red and transformed with YAC will be colorless. Transform with YAC would be colorless. So cloning of a foreign DNA fragment into YAC causes insertional inactivation. So cloning of foreign DNA, cloning of gene of interest in YAC causes insertional inactivation. So the colonies that contain the foreign DNA fragment are red. Insertional inactivation. Let us write here restoring red color. And let us move further, that therefore the colonies containing gene of interest are red. Other thing, that the yeast artificial chromosome vector is capable of carrying a very large DNA fragment up to 2 MB. YAC can carry large DNA up to 2 MB, but the transformation efficiency is low. We'll write over here transformation efficiency is low. Now talking about the homologous recombination in such kind of things. So next is homologous recombination. So in recombinationally targeted yeast artificial chromosome cloning, yeast artificial chromosomes are assembled in vivo by recombination and not by ligation in vitro. So recombination takes place between the target segment of the exogenous DNA and the YAC vector that contain the sequence homologous to these target sequences. So recombination is going to take place. Recombination occurs between target segment of exogenous DNA and yeast artificial chromosome vector containing sequence homologous to these targets. Then the first two yeast artificial chromosome vector arms and the exogenous segments are transformed into yeast cells and followed by recombination. Result is formation of desirable stable yeast artificial chromosome. Now, modifying yeast artificial chromosome by homologous recombination is also possible. Existing YAC clones can be modified by homologous recombination in yeast, and this process is called as retrofitting. So, existing yeast artificial chromosome clones can be modified by homologous recombination in yeast. By homologous recombination in yeast, and this is called as retrofitting. So we'll write in another word called as retrofitting. Retrofitting. Now, what is the uses of yeast artificial chromosome? We'll be talking about the uses. So, uses of yeast artificial chromosome. Uses of yeast artificial chromosome. So, application of yeast artificial chromosome includes generating whole DNA libraries of the genome of higher organisms. So, generating whole DNA libraries of genomes of higher organisms. Then after that, identifying essential mammalian chromosomal sequence necessary for the future construction of specialized mammalian artificial chromosome. So, for identifying the essential mammalian chromosomal sequence, identifying essential mammalian chromosomal sequence. Then another major application of yeast artificial chromosome can be in the study of gene regulation, in the gene expression by cis-acting controlling DNA elements. To study regulation of gene expression by cis-acting controlling DNA elements. Now that are present either upstream or downstream of large eukaryotic genes after the transfer of yeast artificial chromosome from yeast to mammalian cells. That is also the use of yeast artificial chromosome. Now, when we are talking that yeast artificial chromosomes are used for the genomic libraries, let us talk about yeast artificial chromosome genomic libraries. Abdul Rahman is saying good evening, ma'am. Good evening, Abdul Rahman. I hope you are understanding the lecture. Yeast artificial chromosome genomic libraries. Okay, so it is possible to construct the yeast artificial chromosome with mega base long insert using the precise homologous recombination. So, mega base long inserts can be constructed using precise homologous recombination. Then original DNA sequence of eukaryotic genome fragment more than 2 MB in size can be maintained in a single yeast artificial chromosome vector. So very large inserts can be maintained. So that is a very good point about yeast artificial chromosome. So genome fragment मैम हम ऐप परचेज करना है मैम कैसे करें? अब्दुल रहमान आपको गूगल प्ले स्टोर पर जाकर ऐप डाउनलोड करना पड़ेगा। ऐप का नाम है अनअकैडमी लर्निंग ऐप। जब आप ये ऐप डाउनलोड करेंगे और आप प्लस सब्सक्रिप्शन के लिए जाएंगे तो वहां पर आप सबसे पहले तो आप ऐप डाउनलोड करेंगे, आप अपना अकाउंट बनाएंगे और फिर अकाउंट बनाने के बाद आपको ऑप्शन मिलेगा कि आप प्लस सब्सक्रिप्शन लें। तो आप प्लस सब्सक्रिप्शन लेने के लिए जो भी प्रोसीजर है वहां पे अपना नेम और सारी डिटेल्स वो सारी फिल करेंगे। आप मेरा रेफरल कोड देंगे। मेरा रेफरल कोड है FB01। ये रेफरल कोड देने से आपको जो भी प्राइस रहेगा उसका 10% और डिस्काउंट हो जाएगा। तो आप जहां पर भी कॉपी है वहां पर लिख लीजिए रेफरल कोड FB01। ऐप डाउनलोड करिए, अकाउंट बनाइए और उसके अंदर सारी डिटेल्स फिल करिए और प्लस पर जाइए, प्लस सब्सक्रिप्शन के लिए जाइए और वहां पे मेरा रेफरल कोड यूज करिए FB01 और 10% डिस्काउंट लेने के बाद आपका सब्सक्रिप्शन एक्टिवेट हो जाएगा एंड यू कैन अटेंड द क्लासेस। आप प्लस क्लासेस भी अटेंड कर सकते हैं और स्पेशल क्लासेस भी अटेंड कर सकते हैं। अब्दुल रहमान आपको समझ में आया? जीनोम फ्रैगमेंट more than 2 MB can be maintained in a single YAC vector. So as we told that it can carry long sequences. So yes, megabase long inserts such as 2 MB inserts are done in the yeast artificial chromosome. Now there is a comparison between yeast artificial chromosome and bacterial artificial chromosome that we'll be seeing. Abdul Rahman is saying yes ma'am. Oh, yes ma'am. Okay. Hope to see you in the plus classes and special classes as well. Now let us do comparison between the yeast artificial chromosomes and bacterial artificial chromosomes, right? So first feature is configuration. So in today's class, what did we read? That yeast artificial chromosome is having what type of structure? What type of structure it has? It has a linear structure. And bacterial artificial chromosome has got circular structure. Then features. In feature, next thing is host. What is the host for yeast artificial chromosome? The host is yeast, and in bacterial artificial chromosome the host is bacteria. Then the copy number per cell, copy number of such vector per cell. For yeast artificial chromosome it is 1. For bacterial artificial chromosome the copy number is 1 to 2. Then what is the cloning capacity? For yeast artificial chromosome the cloning capacity is unlimited. For bacterial artificial chromosome, the cloning capacity is none, too low. Okay, then what about chimerism? So yeast artificial chromosome shows chimerism up to 40%. But for bacterial artificial chromosome, no data is available. Then what is the insert stability? So here our yeast artificial chromosome, it loses the championship. So in yeast artificial chromosome, the insert has got unstability. Its stability is quite less. And in bacterial chromosome, the stability is quite large. So this we have seen the comparative analysis of yeast artificial chromosome and bacterial artificial chromosome. So like this we have completed this topic in a well systematic manner, and I hope you people have understood the things. So those who are attending the class live, they can write in the chat box whether they have understood or not. And lastly, as we say, let's crack CSIR UGC NET together and GATE 2021 together. If you haven't subscribed our channel on YouTube, subscribe it now. Hit the bell icon for getting the notifications. And if you really like my video, give it a thumbs up. And for plus subscription, use my referral code FB01 for getting a 10% discount. You can also attend my special class and special classes of other educators by using my referral code FB01. Every day 10:30 in morning I take the classes for CSIR UGC NET, and in evening 5 o'clock I take classes for the GATE. So I hope to see you tomorrow morning at 10:30 a.m. for CSIR class, and in the evening 5 p.m. for GATE class. Lastly, thank you very much for your patient listening. It was great teaching you, and I hope to see you in other classes as well. Till then, be happy, keep on studying, and have a very, very wonderfully good day. So, it's a finally bye from my side. Take care, study, and those students who were asking me how to get the plus subscription, how to get the app. So, Abdul Rahman was asking me. I hope the things are quite clear, and go for purchasing the app. It can really be a what to say milestone in cracking the examination, because all of the things which you haven't read in your college examination or college classes, you can actually read over here, because all the things are available in a well systematic manner. So go for the plus subscription. This is the time, this new year is the time to go for it and achieve whatever you want in life. It's a great best of luck from my side to each and everyone. We'll be talking about flow cytometry in the morning class, and we'll be also doing an evening class tomorrow. So Prabhat Kumar is saying good evening ma'am. Prabhat, you are a little late for the class, but do you have any kind of doubt or query? Because my lecture, I'm just going to sign off. So do you have any query? So you can write, otherwise you can watch the whole video since starting. I've taught yeast artificial chromosome. So Prabhat, are you having any kind of specific doubt which you want me to clear? I'm waiting for you to answer. So I think it was a general wish from a student to a faculty. Thank you for your wishes, and we'll be meeting tomorrow morning and tomorrow